I will be speaking on individual variability and brain evolution. This presentation was part of the educational course on brain parcellations and functional territories at the Organization of Human Brain Mapping Conference in 2020. In order to define functional territories, we need to parcellate the brain within a common anatomical framework. As a result, parcellation methods use group averages to make individual brains comparable. While using a common anatomical framework, or template as it's often called, is helpful to compare people to each other, it is important to remember that we are all different. If we take a non-brain example here, we all wear shoes, and the average size for a woman is 8.5 in the US, 38 in the EU, and 6 in the UK. That variability in itself shows you how different we are. Now imagine putting on the average shoe when you aren't the average size. Walking through life would be very difficult in that case. However, when it comes to the brain, we tend to do exactly that. We put the same shoe on everyone. This is in part due to the fact that we don't have a magic number yet of how different we are, and it's easier to start from a common ground. But knowing how variable we are is important, because we need to explain how the brain functions and dysfunctions on an individual level. At the moment, we build brain models on an average template. Based on these models, we then create treatments in medicine. But when we apply these models in the clinic, they often don't fit the patient, rendering the treatments ineffective for some, but not all patients, or even have unexpected side effects. This one-size-fits-all method is what we have done in neuroscience for a very long time. And while the fashion industry has addressed that we need different sizes for different people, neurosciences are just now coming to terms with this principle. So the question arises, why are we different? Looking at nature, we can see that the variability stems from and may drive evolution. Let's listen to Neil deGrasse Tyson, who explains it best when talking about the evolution of the eye. So here we see that the complexity of the eye makes no sense without evolution. But the brain is a highly complex organ as well. So what is the role of evolution in the complexity of the brain? And does variability in brain shape and function lead to a cognitive advantage? Evolution means variability, as we have just seen, and that variability provides an advantage of some sort. So what do we know about the variability of the brain and the cognitive advantage that might come with it? On the top left, you can see a study looking at creativity and showing the different cortical areas that can be associated with it. On the top right is the first study that looked at the variability in the frontal-parietal white matter network and the impact on pseudo-neglect in healthy volunteers. In the next study, we demonstrated that white matter variability is clinically relevant when looking at the recovery after stroke and symptom severity in neurodegenerative disorders. Finally, in order to study the evolution of the brain, we need to study interspecies variabilities. The ever-growing field of comparative anatomy studies has shown that other species have different brains when looking at the surface or the connections of the brain. While comparative studies have been an integral part of brain mapping for decades, only with the emergence of MR imaging has it been possible to study the same methods across species. Why is comparative neuroimaging useful? Well, to begin with, we don't have to sacrifice our study participants. Traditionally, comparative studies have been based on post-mortem measures such as histology, blunt dissections, and axonal tracing. While some of these methods can be applied to the human brain, they are by nature only possible ex vivo and therefore offer limited comparability. In recent years, though, in vivo MR imaging has become a useful tool for a direct comparison of structure and function between species. For our study, we wanted to use cutting-edge in vivo imaging to compare human and macaque brains and map interspecies variability. In order to compare how different we are, we first created a template that represents the group average for the grey and the white matter. We then compared how far every single individual is from this average. Through various iterations, we can see that the best numbers we can reach for shared variance is somewhere between 80 to 100%. So now we know the percentage of shared variance. Next, we wanted to know how many individual brains it takes to build a reliable or representative template. As you can see from the chart, it takes about 10 people to build a template for grey and white matter maps. It is possible to add more people to a template, but percentage of shared variance will not considerably improve. Now that we have our reliable templates, we can go ahead and compare how different our brains are to each other. However, one thing to consider before is that we are comparing monkey and human brains. One obvious difference is that monkey brains are smaller in volume. So does size matter when it comes to variability? The average monkey brain is about 1/12 of the human brain, or for a more tangible comparison, we are comparing biscuits to pizza. When comparing monkey and human brains, we have two measures are available. One is the absolute distance or Euclidean distance, and one is the proportional or Jacobian deformation. When considering Euclidean distance, we can see on the left that the human brain is more variable for grey and white matter, while the monkey brain is less variable. If, however, we look at the proportional deformation on the right and thereby account for the difference in brain size, the variability in human grey and white matter is comparable to the monkey grey and white matter. This means that overall, the monkey brain is structurally as variable as the human brain when brain size is controlled for. Now let's map the variability on the brain. Looking at the grey matter surface, both in humans on the top and macaques at the bottom, we can see that there are areas of high and low variability. The warmer the color, the more variability there was for this region. When looking at this pattern, we can appreciate that areas like V1, V4, the parieto-occipital sulcus, and superior temporal sulcus are some areas of high variability, while areas like V2, V3, and the ventral prefrontal cortex show lower variability. This pattern might indicate that areas of high variability may have evolved more recently and be less similar across individuals. This means that some areas are more different than others, both in the individual human and monkey brains. And this has a big implication when we are studying atlases and functional parcellations of the brain where such differences are commonly unaccounted for. The anatomical variability on the lateral surface appears to follow evolutionary trends as described by the dual origin theory. According to this theory, brain evolution emerged from two systems, the hippocampocentric and the olfactocentric divisions. These systems meet in the more recently developed regions located in the middle frontal gyrus, temporal lobe, and inferior parietal lobule. These converging areas have been associated with decision-making, language comprehension, and tool manipulation. We also found these converging areas to be the most variable in terms of their anatomy, which seems to follow the gradient suggested by the dual origin theory. Our maps therefore indicate that variability of the brain may be linked to evolution. Using MR imaging measures in humans and macaques, we can report the first evidence of a relationship between anatomical variability and evolutionary expansion maps of the primate brain. When looking at the evolutionary expansion and variability, we can see that some lobes of the brain have a positive or negative correlation between the two, for example, in the temporal, limbic, and occipital lobes. These patterns are similar between both hemispheres. Looking at the two hemispheres, we can also study the asymmetry in variability between species for separate brain regions. Overall, areas of greater asymmetry are mainly those with the largest degree of hemispheric dominance in humans. Whilst we have seen that the monkey brain was overall structurally as variable as the human brain has an additional left hemispheric asymmetry in variability across several phylogenetically recent regions. Most of these regions are associated with higher cognitive functions, such as language, for example. The strongest asymmetry can be seen for the left temporal lobe in the human brain, while the inferior parietal lobe was the only region in the monkey brain that showed asymmetry for variability. So far, we have looked at the variability of the grey matter and individual grey matter areas. Next, I want to talk to you about the white matter. Here I'm presenting to you the first map of white matter variability. We quantified for every subject the average white matter deformation in each voxel and projected the results on the average stratigraphic reconstruction of the human and the monkey brain. Overall, the superficial white matter is more variable than the deep white matter. For the superficial white matter, the pattern is similar to the grey matter variability maps. We again see the same trend whereby older parts of the brain tend to be less variable between individuals, while areas associated with higher functions tend to be more variable between individuals. Here, I'm presenting to you the first map of white matter variability. We quantified for every subject the average white matter deformation in each voxel and projected the results on the average stratigraphic reconstruction of the human on the top and the monkey brain on the bottom. Overall, the superficial white matter is more variable than the deep white matter. For the superficial white matter, the pattern is similar to the grey matter variability maps I showed you just before. We again see the same trend whereby older parts of the brain tend to be less variable between individuals, while areas associated with higher cognitive functions tend to be more variable between individuals. We have seen that evolution has played a role in the development of the brain. We have seen that monkey brains are structurally as variable as the human brain when brain size is controlled for. Within species variability may be another indicator to study brain evolution, and areas supporting higher cognitive functions tend to be more variable. We also have seen the first evidence of white matter variability within and across species, and going forward, the parcellation methods we are using today are helpful when looking at older structures of the brain, but for more variable regions, we might need to reconsider our methods. We need to find a way to parcellate the individual brain in a reliable and comparable way across subjects, both for the grey and the white matter. So, when it comes to the brain, one size does not fit all. And if you want to learn more about this topic, I invite you to read our paper in cerebral cortex. Many thanks for your attention.